Good evening, everybody. Okay, I'm going to do this really fast. I'm going to basically do a review of all 4 of the different lab portions that you guys had to do, basically because it's a 10-minute video that I limit to on YouTube. So the first part we're going to go through is plasmids, and then we're going to go through restriction enzymes, gel electrophoresis, then PCR, then transformations. If I leave anything out or forget anything that you need explained, make sure you ask in the comment bar down below, and I will make sure to get back to you and reply in respective comment bar so that everyone else can see it as well, or do another video to respond. So we'll go ahead and start with plasmids. So plasmids are very useful in recombinant DNA engineering. The main reason being is because they're easily workable. They, on their own, they can be, in real life, they are actually very useful. So if we have 2 bacterium and then each of them has their own circular chromosomal DNA, in addition to that, they'll have little plasmids. These little plasmids, because of the universal DNA code, can work in either bacterium, which makes them very useful because you can take something from one bacteria and have it express itself in another bacteria. Extending that even further, we can actually take eukaryotic DNA, such as our tomato gene of interest here, or our red fluorescent protein, RFP, which actually has come from a jellyfish, so it's eukaryotic. We can place that eukaryotic DNA fragment into a plasmid, which is bacterial, and then we can actually put that into a bacteria and have it expressed there. So you have a eukaryotic protein expressed in a bacteria, which is actually pretty amazing. Now, there are several things that make it very useful, in addition to be able to introduce foreign DNA into a bacterium, and then using that, taking advantage of that universal genetic code. They replicate themselves independently of this chromosomal DNA right here. In doing so, we are able to make many copies of that little thing without ever having to worry about the overall regulatory processes of that entire bacterium. So this bacteria can also transfer itself into other bacteria. This plasmid can transfer itself. And we actually will do that in lab, and you already have. Recombinant plasmids are useful for several reasons. We have here our recombinant plasmid arabinose, which has the arabinose operon in there, and what happens is arabinose, which is a sugar, will bind to RSC. RSC will kind of bend over and allow for DNA polymerase to get captured and placed onto this promoter region, and it'll actually just go ahead and transcribe this tomato gene here, or the RFP. This red fluorescent protein will then be expressed. What is also usually present in a recombinant plasmid is some kind of selection marker, which we use during our transformations. The selective marker is always on, so it's constitutive. Right? And this allows us to select later on plating on ampicillin. So this ampicillin resistance creates a protein which will actually be excreted by the bacterium and allow it to be resistant and able to live in ampicillin, which is an antibiotic, and antibiotics usually kill bacteria. So this is really important. It's a selective marker that we'll be using in the transformation section, which I'll explain more about it then. Restriction enzymes can also be cut at very specific sites. So here we have our Hindi3 and our BAMH1 restriction enzyme sites. And this allows us to actually insert that tomato gene or that RFP gene, which is very handy. And what the restriction enzyme does is it cuts the phosphate backbone. So we're going to go ahead and take a look at restriction enzymes really quick. Let me clear up my previous blackboard. All right. So what we have here are one of the ones of interest. We use Hindi3 in lab, and basically what it allows us to do is create some sticky ends. So how restriction enzymes work is that they kind of cleave the phosphate backbone, and they create palindromic sticky ends, or little like overhangs. And these overhangs are specific. So we have AATC in the 5' to 3', and again in the 5' to 3', AATC. And then here for the Hindi3, we have AGCT, AGCT. So these sticky ends allow for specificity in the rebinding, which we use with ligase, and allows us to insert things into our restricted fragments later on. It also ensures for directionality. So if we can go back to what we have here, if I had used a blunt-ended restriction enzyme, sorry about that. If I had used a blunt-ended restriction enzyme, which is a little less specific, and then I religate it in, what might happen is that this tomato gene might actually glue itself back in the backwards order, which can actually be rather damaging if you think about it, because then that tomato gene might not work so well anymore. So just keep that in mind. All right. Moving on, restriction enzymes are endonucleases. They cut the phosphate backbone, and if they're non-specific, what you will have oftentimes is something like a SMO1 restriction enzyme, which actually just pretty much cleaves right down the middle. So this is still palindromic, but it's non-specific. It doesn't have a sticky end. It can pretty much just ligate onto anything else that has a blunt end. These sticky ends are also called cohesive ends, so keep that in mind, and they can be ligated together. And then in real life, the purpose of restriction enzymes, one of the purposes they serve amongst many, is to prevent a bacteriophage from lysing or eating up or tearing up that bacterium. So it actually serves a really good purpose in real life. But in the lab, we use it to actually help us engineer DNA. Okay, so moving on, I'm halfway through the 10-minute video. Gel electrophoresis. I'm going to try and do this even faster now. So gel electrophoresis is basically just taking advantage of the fact that the phosphate on the DNA backbone is a negative charge. So that negative charge is going to be repelled by the negative charge on one side, which we're going to line up our wells, and it'll be attracted to the positive charge on the other side of our gel that we run. One way that I remember it is run to red. So as long as you remember that you're going to run in the direction of red, you know that red's going to be on this side. If red is on one side, black must be on the other side. Black is usually associated with morose, negative kind of emo, so you can think negative. So if this side is negative, this side must be positive. So run to red. The other side must be black. If black is negative, then the other side must be positive. That's how I remember it. One thing you want to keep in mind is that larger things go slower through this agarose gel matrix and will end up nearer towards here because they travel slower. Smaller fragments or smaller conformation fragments of things will move faster and will thus migrate further along towards the other end here. There are a couple things that you use. We have a tracking dye. So our tracking dye that we use is bromophenol blue, and this helps with helping you see that, yes, I did load it into the well, and helps you see how far along the migration is going when the electrodes are running, so that you don't accidentally run your sample off the gel. We have our loading dye, which is a glycerol or sucrose, and it weighs down the DNA sample. So the DNA will actually stay in that well and not float around and everything that's out here. Ethidium bromide is a stain intercalator. Basically what it does is it inserts itself between the base pairs of the DNA, and when you put it under a UV light, you'll be able to see here basically a picture of where your DNA fragments are. Your visual tracking dye is not able to do that. What we have here is an example of our restricted cut plasmids. So we have our larger fragment here, and then here we have our little tomato gene. And this is positive because, yes, we have done a restriction here. This is our ladder, our marker, or our standard. Those are the three names that are usually used to describe it right here, this box. Let me see if I can get back to the pen. And then what we have in this little box here is one that's unrestricted. This unrestricted blur of multiple bands, what you're actually seeing is various states of conformation. So a large non-folded state will travel slower, and then something that's really clumped up will move really quick, and then something that's maybe like only partially clumped up will move medium speed. So that's what you see in this unrestricted plasmid region here. Finally, we want to talk about PCR. I am not really going to talk too much about PCR. Instead, I'll link a video on it. Basically what you're seeing is you're going through methods of denaturing, which usually happens using DNA helicase, but PCR uses heat, then annealing, then extending. One thing you really want to be prepared for is that, yes, you use helix to prep your DNA, but Taq polymerase is the main thing. This Taq polymerase is actually found from a bacterium that's very thermostable that was found in Yellowstone Park there, and it's magnesium chloride sensitive. So PCR will copy one specific gene over and over and over again, which is very handy. So here we have an example of the ALU gene, which you did in lab. This was my actual sample from when I did it a long time ago. Sorry, my cell phone goes off like that. So we have our ladder. We have a prep lab worker who volunteered their DNA over here. A contamination control, which was just H2O or water, and there should be no bands here, which shows that we had no contamination. Me personally, I have ALU on none of my chromosomes, so I have a negative here. This person here, Stephanie, she had it on both of her chromosomes, so she showed one band here. And then David, my other lab partner at the time, had one on one of the chromosomes and then not on the other, so you show two bands. And Dr. Hyder Haidar actually used her DNA as well as a control, and she had a plus-minus expression as well. So that's just what you're seeing here. It's very straightforward. Make sure that you compare a proper ladder to here. So if I use a ladder that was too dense, I might not be able to see that, yes, this lines up with the 400 base pair and 100 base pair for the smaller one. Okay, so moving on again. Sorry about my phone. I'm getting a lot of text messages all of a sudden. So we have transformations. Basically in our transformations, there are a couple things that you want to be aware of. We used heat shock to maximize the uptake of the plasmid through the bacterial walls. And then we also, our transformation solution that we use, if you remember, is calcium chloride. And then afterwards, after all the heat shock, these poor little bacterium, or actually these E. coli, are really in shock. So we need them to heal, and we give them nutrient luria broth. So after they heal, we plate them and they grow. We come back the next lab session and we find 4 different plates. All right. So one plate just has luria broth with non-competent E. coli. These non-competent E. coli did not pick up, so both of these plates did not pick up that plasmid with the ampicillin resistance. All right. So what we have here is luria broth. No ampicillin, so everything will grow on this plate. Any and everything that hits this plate will grow, basically, all the bacterium. So what we see is a lawn of everything. And this is the E. coli that non-competent. Now, we also have non-competent E. coli, which means it didn't pick up a plasmid with that ampicillin resistance on a luria broth plate. So there's nutrients, but there's also ampicillin, which will kill off, which is an antibiotic, and it kills off all the bacterium. So you shouldn't see any colonies there. If you look at our plate with colonies, you'll find that those are competent, or they did take up the plasmid DNA with the ampicillin resistance. So they are able to live through the ampicillin. So these 2 plates are the same, except for the fact that this one does have competent cells. And the luria broth is a nutrient. So these guys will form colonies, and these little colonies, great. You have colonies that do form. That's wonderful. If you look at your other side, this one also had competent cells. That means they did take up an ampicillin-resistant plasmid and produce ampicillin resistance. And this ampicillin resistance is basically allowing these little colonies to form again. But in addition to that, we have arabinose. Now, if you remember from our original plasmid, if I could just move this up front, our original plasmid here, we have the protein activator, RSC, which binds arabinose, which allows the promoter, which allows for everything to be expressed. So red fluorescent protein is only expressed in the presence of arabinose that binds to the activator, which allows the promoter to do its job. So if we go ahead and take a look again back at here, what happened is exactly that. With arabinose present, it binds to the protein activator, which captured the DNA polymerase, allowing the production of the of the tomato protein. So this RFP started to express itself. When we turn off the lights, yeah, they glowed in the dark. It was kind of amazing. So you can see what different conditions will lead to different things. Stephanie asked a question before, and I explained it, how you can tell competent versus a non-competent cell. I'll link that video right on top of this screen as well on YouTube so that you can get a good view of that. If there are any questions that I did not answer, please let me know. I paid close attention to you if I were you to all of the stuff that was used. So like calcium chloride and heat shock was used in transformations, in restriction enzymes. If you notice there, which restriction enzymes, why we use them, because they're specific. In gel electrophoresis, you really want to pay close attention to the fact that we have a loading dye, that we have a tracking dye, what's the difference between them, what's the purpose of ethidium bromide and agarose, and then in PCR, you want to be very cognizant of the fact that we're not using regular DNA polymerase. We're using Taq polymerase, which is very thermostable and magnesium chloride sensitive. And you want to make sure not to mix up magnesium chloride used during PCR with the transformation calcium chloride. That's one common mistake people might make. But if you have any more questions, put them in the comment bar, and I'll do my best to respond in a timely manner, either through video or, again, by responding to that in the comment bar again. All right, good luck, everybody. I hope I didn't talk too